Hi, my name is Rebecca Taylor, and today I'm here to tell you about work that's been going on at Carnegie Mellon University. My team, Shriram Kumar, Alexander Pierce, and Ying Liu, are really responsible for all of this work, and they talk with me about it a lot. So I want to give them proper credit up front. So the topic today is probably a very familiar approach for many of you, but with a somewhat unusual material. So we've titled this Self-Assembly of Nanotubes from Distinct Gamma-Modified Peptide Nucleic Acid Oligomers in Organic Solvent Mixtures. All right. Now I want to take a moment just to introduce my group. And so we are the Microsystems and Mechanobiology Lab at CMU, the MMBL, or the Mumblers. And so if you would like to find out more, please check out this link, or you can just search CMU and MMBL, and you'll find us. Briefly, a little bit about me. Who am I? So I'm an assistant professor in mechanical engineering. I have a courtesy appointment in biomedical engineering, electrical and computer engineering, which really just says I work across disciplines and also across scales. Fantastic. Now, I've been at CMU since 2016, and in my lab, we have three major thrusts for our research, and that includes cellular and molecular biomechanics, bio-inspired micro and nanosystems, and biomanufacturing and nanomanufacturing. And this project fits into two of these categories. We're inspired by gamma PNAs as attractive materials for building bio-inspired nanosystems. Further, we're excited to understand the science of nanomanufacturing with gamma PNAs, which enable the fabrication and utilization of nucleic acid nanostructures in environments that typically would lead to degradation of structural DNA nanotechnology. Okay, so as an overview, there are really 6 items that I want to talk about to introduce our work. And so to begin, I just want to tell you a little bit about gamma-modified peptide nucleic acids, or gamma PNA, as we'll refer to them. And it's a really interesting and exciting material. So peptide nucleic acids bind strongly to DNA and RNA. And in fact, when they are modified at the gamma carbon position, this can lead to pre-organization of the peptide nucleic acid, so basically making it chiral, reducing its conformational flexibility, and further enhancing binding, right? Gamma PNAs and PNAs resist degradation by nucleases and proteases. They're slightly different than DNA. And so in terms of twists, so if you look at the picture on the left, you can see that it takes about 18 bases for PNA or gamma PNA to undergo one full rotation. And what does that mean? Well, that means that it's less twisted than DNA. So if you were building a nanostructure out of PNA, some of the rules are going to be a little bit different, right? Other things that are really exciting are that because PNA binds strongly to DNA, or binds very strongly to DNA and to itself, you can use as few as 5 bases as a recognition sequence. And this has been previously published. And so I should say at the bottom, I have some links relevant to the development of PNA by Nielsen, as well as recent papers talking about specific gamma modifications on peptide nucleic acids. Oh, and I think the last thing is that PNA can hybridize in organic solvent mixtures. And so situations that might lead to a DNA nanostructure falling apart, those might not cause a problem for PNA. Okay, so specifically to get into the chemistry, here you have your nucleic acid labeled as B, and you can see that PNA and DNA are quite different. So PNA actually has an amino-ethyl glycine backbone. And so what this means is that inherently it's uncharged, but it's got similar axial rise to DNA, and it can be modified with the same nucleobases that DNA can. Now, introduction, like I said, at that gamma position of modification can lead to global changes in organization of the PNA molecule and can also give it just a dramatic amount of versatility, right? And so Sahu et al. Has described this in detail. And so I would urge everyone to check out this paper if you're not familiar with it. Also, this diethylene glycol as the modification, which I'll refer to as mini-PEG from here on out, is very useful as a molecule, but there's many functionalizations we could use. In this work, we were working with mini-PEG. And our hypothesis was essentially that nanostructures, complex nanostructures, could be built with modified PNA. Okay, so to introduce the decision-making process that we went through, I first want to talk about what sort of nanostructure can we build, right? And so we're all familiar with DNA origami, but we're also familiar with single-stranded tile systems. And one of the major benefits of a single-stranded tile system is that it's really, really easy to make large structures from very, very small sets of oligomers. And so one of the first constraints that I want to make everyone aware of is simply that we didn't have unlimited funds. So given the expense of making PNAs and gamma PNAs, we worked with a small number of short sequences, or that was our goal. It doesn't mean this couldn't be expanded to an origami-like approach, but at this point, we've really just scratched the surface of investigating more nanotube type structures. So periodic nanostructures made of small sets of short sequences, each of which has multiple domains, right? All right, so I'm sort of already getting into this, which is, what are those constraints that you would need to deal with if you wanted to make a system that could successfully form out of PNA? And so the first, obviously, is cost. The second has to do with the length of the PNA, okay? And so I mentioned that 5 PNA base pairs are sufficient, or been published to be sufficient for recognition sequences. So our first thought was, instead of using a 10 or 11 base recognition domain like we oftentimes see in a single-stranded tile system, we wanted to see how short we could make that recognition system. Okay, and so what I'm showing, and I recognize that this may be a little confusing at the top, but at the top we have 9 strands that are each 12 bases long. And what I want you to understand is that each one that binds to another only binds with 6 domains, right? So if P2 and P3 bind together, 6 bases are interacting. If P2, P3, and P7 are binding together, that means P2 and P3 bind with 6 bases, and P3 and P7 bind with 6 bases. So we could imagine taking lots of short strands to build a nanostructure, but we wanted to verify if that would be sufficiently stable. So running melting curves in both PBS as well as DMF and DMSO, we found really supportive and interesting results, right? So just 6 bases of P2 and P3 gave us a melting temp of about 39 degrees. And when we added in P7, we saw a cooperative rise up to 59 degrees for that melting temperature. So for those curves, that's the solid orange to the solid black. And now the other thing that's interesting is if we're using that solid black line as kind of a gold standard, what we see when we replace PBS with DMSO is only a minor reduction in melting temperature down to 54. And even in DMF, we see a minor reduction from that. So it potentially is feasible to think about select organic solvents as the solution in which we form these nanostructures. And this work is forthcoming in Nature Communications. And I want to also take time to point out the majority of this work has really been the focus of Shriram Kumar's work during his PhD, and so I have his picture in the right corner. And his, dedication to this project has been fantastic, and his insights into how to work with peptide nucleic acids and learn the lessons about how they're similar and different from DNA has been fantastic. So if you are in the future looking for a postdoc, I want to recommend you reach out to Shriram Kumar. Okay, so this is the structure that we've been building. And this is why I showed the 9 distinct strands on the previous page. What we actually came up with was a simple repeating structure. And we wanted to call it a quote unquote nanotube with a minimal number of helices. So it's really just a tight bundle of helices. And in this bundled structure, we would use the 9 oligomers that were each 12 bases long to basically form a ribbon, and that ribbon rolls up on itself. And the reason we did that was we wanted to get as short as we could with our PNAs. Practically speaking, the yield of PNA goes down the longer you get, and somewhere around 20 to 21, 22 bases is about as long as practically can be synthesized at present. And so we wanted to limit ourselves to very short PNAs if possible, so we only used 2 domains that were each 6 bases long. So those are very short peptide nucleic acids. And furthermore, if you've got 2 domains, you do not have even a 2-dimensional structure, right? A binds to B, B binds to C, C binds to D. That's linear, right? But because it rolls up on itself, we get this 3-helix nanostructure. Okay, great. Oh, and what you see also on top is that we've introduced our gamma modifications. And so at this point, the gamma mods are indicated by these small little circles. And basically, these are going to pre-organize the structure and increase the binding affinity. And our hope was that this gamma modification would enable the structure to form and remain stable. We also avoided having gamma mods at these crossovers where our strand would bend back between 2 helices. And if you would like to ask more questions, I'm happy to connect offline. Okay, so because sometimes it's difficult to visualize what's happening without a video, I thought I would play a video. So here we have our bundle, and you can really see how it's knitted together, with the green strands acting as what we call crossover strands, forming that link between each of the helices. Whereas the cyan and magenta strands, we call them contiguous, because they stay on the same helix for the entirety of their function in the structure. All right, so these are our 3 helix bundles. And if this is DNA, if this were a DNA type system, we would hope to see structures like this. Okay, so then let's talk about what we found. Now, we found that we can form these structures in 75% DMSO and 25% water, which means we actually had to adapt many of our processes that we typically perform in a more water-based environment to accommodate the DMSO. Certain things like degradation in microscopy results, or at least absorption from DMSO causing challenges with microscopy, what is an interesting part of this work. But we were able to show, using total internal reflection fluorescence microscopy, that we can build filaments that can be as long as 11 microns. And so the scale bars on these plots are 5 microns. And what you can see is that indeed they're growing somewhat like you would expect from an SST system. There may be some branching here that we're interested in, and there are a lot of like short particles. We can't tell from this turf microscopy whether these are short filaments or whether these are aggregates or what's going on there. So we excluded all structures shorter than 2 microns when we were doing our length measurements, so that we could be more certain that this was actually filamentous in nature. Okay, so but I think you really want to look closer, right? So anyone that's worked with SSTs would also probably be a little questioning of the previous result, because they just looked sort of fat. And for a structure that you might expect to be 6 nanometers in diameter, you don't expect a very broad filament. Far less than a pixel in size would be my expectation. So those look unusual, even though indeed they're filamentous. And so TEM imaging revealed that indeed we have some strange things going on. Okay, so to start, we have branching. I can say the scale bars here are 100 nanometers. And so indeed, these are very wide. And beyond that, this is not a monodispersed population. So this is very surprising to us, is that we see a distribution of widths, right? So there are either two things going on here. One, formation is not occurring as we would expect, and these structures are interweaving to sort of come together into these global bundles. Or perhaps they are more adhering together and aggregating, and that aggregation is part of this process. Okay, and so at this point, we can't tell from the TEM. What we can see, which is encouraging, is that we have a banded structure across the width of the filaments. And so here I show the normalized stain intensity, and you can see that these tight bands have something like an 8 or 12 nanometer period. And that's consistent with being constructed of sort of a bundling of our structures, which we expected to be around 6 nanometers. All right, now much of this work really involves adapting all of our techniques to work with solvents that we don't typically work with. And when we talk about solvents, the thing that's exciting about DMSO or DMF is that these are solvents that are critical in polymer synthesis, peptide synthesis. And so one exciting avenue for work like this would be to create nanostructures that aren't cross-linked. They're still active, right? And they're not completely encapsulated. That could potentially be responsive materials that could both form and operate under what previously I would consider harsh conditions for a DNA nanostructure, right? But what it meant is we have to adapt all of our techniques. And so here is what we do to prepare our turf studies. And we're putting together a methods paper to provide the community with further detail on how to do this. But I think the first thing to point out is by making these filamentous nanostructures and having them be microns long in length, that's one achievement. But one thing we're also really excited about is we have in our set of peptide nucleic acids, we have one that's biotin decorated, and we have one that's labeled with a fluorophore. And so what this means is that we can use that biotin for attachment. And clearly, because we did turf studies, we can use that fluorophore to do fluorescence microscopy of these nanostructures. Okay, so here's the flow path, which basically involves incorporating Trolox and washing with PBS after depositing streptavidin, and the biotin BSA is the first step of our sort of sandwich, right? So biotin BSA, streptavidin, nanotubes. Okay, so let's move on to the next part. So one exciting thing about DNA and PNA is that they can bind together, right? And so we wanted to explore to what extent we could incorporate DNA into our gamma PNA nanostructures. Okay, so this slide I'm going to leave up for a while, and then I'm going to talk about it, and I'm going to come back to it in a little bit. And there's a lot going on here. We have turf images in column 1 and 3. We have TEM in columns 2 and 4. And as you go down from top to bottom, we increase the amount of DNA that was incorporated into our PNA structures. So what I would have expected at this point would be that you would get a dose-dependent change in the morphology, depending on the amount of DNA that was incorporated. And we don't see that. The images from top to bottom look very similar, right? So this is one thing. The other thing is that on the left, we talk about replacement with a contiguous strand, right? One of these strands that remains on the same helix the whole time in the structure, versus the replacement of a crossover strand, one of the ones that turns and goes back and sort of spans two helices. And so these look like completely different structures, right? So there's a real question of why is this happening? You know, and we find this fascinating. And at this point in time, we don't know. I can tell you what we were expecting to start. So we've been very excited about previous studies measuring the basis per turn for various constructs, whether they're PNA-PNA, gamma-PNA DNA, PNA-DNA, and DNA-DNA. And the trend I want you to see here is that PNA-DNA systems only have about 13 bases per turn. Gamma-PNA DNA systems have about 15, right? So we would expect that our regions that are all PNA would have 18 bases per turn as their preferred crossover spacing to be unstressed or untwisted in solution, whereas gamma-PNA DNA would be more twisted. So we thought the twist was going to play a big role. And it actually seems to be kind of the opposite. So going back to this, I was looking for twisting, and I don't see it. So what I do see is a structure that looks, what I would say, it implies that some sort of pre-stress stiffening is occurring. And we don't understand exactly what could have led to that. But we don't from our bands, from the dye that's accumulated at the edge of the filament, we don't see twist. And it may be there, but it's not obvious. So future studies, for these future studies, we want to look into whether this effect is happening or what is this surprising new stiffening that could be potentially very useful. Likewise, it's really interesting that incorporation of the crossover strands leads to a more like kind of stellate-like structure and much more amorphous morphology with almost like an unraveling type look. So it really speaks to the fact that we've already shown the modification is something we have control over and is critical, but also the design of what I would call the structural motif, the way your structure is interwoven, is clearly critically important. And so in our next steps, we're going to be looking into this. Okay, so one thing I want to say is, I just said that the modification is important. Now, previous studies had shown that incorporation of too much modified or unmodified PNA can lead to aggregation. So there were limits to complex nanostructures being built with PNA and DNA. But we'd like to actually provide evidence of that. So what we did was we did an experiment where we started doing substitution, not with DNA, but actually putting in this unmodified PNA. And what we can show is that it occurs that our structure can tolerate about 4 of the strands being unmodified PNA before we get this switch to just everything aggregates, right? So it's both dependent on the solution as well as the ratio of the constituents. But in general, what this suggests is that there's really something special about the gamma modification of the PNA that is enabling it to form these dispersed structures, right? And so we've chosen the mini-peg modification, and if it's a hydrophilic, hydrophobic issue, we'd like to study different modifications and also play with the length of the domains and sort of the structural motif in general. So, so these are next steps for us. But in general, there are really unique opportunities for something like a gamma PNA for making structural nanotechnology that can be DNA free or a hybrid. Okay, and then I think the last thing I want to talk about is that aggregation is a huge problem, right? And so it may be the case that PNA nanostructures are really exciting because they don't act like DNA. but certainly we want to know how to make them act like structural DNA nanotechnology. So this open question that I want to address last is, what can we do to make these structures avoid aggregation problems? Okay, and so for our first study, what we did was we introduced SDS during the anneal. And so what I want you to follow is that with 0 SDS, we can get filaments like we already saw. And as we slowly increase the SDS, filaments appear to get finer and finer. And at 3.5 millimolar, finer and finer. And at 5.25, they're so fine, you can barely see them. And in fact, these are total internal reflection fluorescence microscopy images. So these are the 5 micron scale bar images. But this is the sweet spot. We also see some interesting behavior if we add too much, kind of creating a network type effect. But we looked at our sweet spot, this 5.25 millimolar. We found that we got filaments that looked much more like what we'd expect, right? So here we have a plot showing the width of the gamma PNA filaments. And we can show that that distribution tightens up substantially and shifts down as we add sort of the critical level of SDS. So SDS is apparently one way that we may be able to modulate aggregation in a system that contains PNA. Because understanding how gamma PNA nanostructures can be like DNA and mimic them is critically important. But I think also the flip side of that is gamma PNA nanostructures may just act more like peptide systems. They may act more like proteins. And so it might be the case that we can be excited to leverage that, but we need to also learn strategies to modulate that behavior. So going forward, I've introduced gamma-modified peptide nucleic acids, the geometric considerations that you are required to start working with them as a building material. I've shown you the filamentous nanostructures that we've formed in organic solvent mixtures, talked about hybrid structures containing both DNA and gamma PNA, as well as hybrids where you have gamma PNA alongside unmodified AEG PNA. And finally, I've talked about the mitigation of aggregation using a surfactant. So what's next in summary is, we really want to understand this apparent stiffening mystery. Mysteries are always the most fun and clearly have some really fascinating physics behind them. We also want to understand the size dependence of aggregation effects. We are really excited to be able to make a big structure, but we wonder if a fixed bounded size small gamma PNA structure would suffer from the same aggregation issues. Will it act more like DNA if it's small? And so we'll be looking into this. Once again, the density and type of gamma modifications is a really rich area for investigation. So we are delighted to be part of the CMU community that's pioneering many of these new gamma modification approaches. And then finally, we're looking at applications. So can this be used in manufacturing, in polymer engineering, or in peptide self-assembly? Because now we have a material that may be more predictable than, say, an all-peptide system that might be able to help us with peptide self-assembly and maintain stability in an organic solvent environment. And so with that, I want to say thank you so much. This is my lab, and I've highlighted Shriram and Ying. Shriram, this has really just been a huge effort. It's been exciting to work with him on this. I want to thank my collaborators and my funding sources. And I really appreciate your time, and have fun at the rest of FNANO.